Hello students, this is the most important question series of chapter Biomolecules and this is the part 3. So we will start with the question number 11. Select the option which is not correct, we should underline here, which is not correct with respect to enzyme action. With respect to enzyme actions. So first option, that is substrate binds with enzyme. Substrate binds with enzyme as its active sites. B option, that is addition of lot of succinate does not reverse succinate does not reverse the inhibition of succinic dehydrogenase by malonate. And C option is a non-competitive inhibitor. Non-competitive inhibitor binds the enzyme at a site distinct from that which binds the substrate. And D option is malonate is a competitive inhibitor. Of succinic dehydrogenase. So here important thing is not correct with respect to, so addition of lots of succinate does not reverse. Really, when addition more substrate is there, automatically substrate concentration increases, so this should be reversed. So further this is not correct, does not reverse, so further this option is the not correct. Then question number 12. Transition state structure of the substrate formed during an enzymatic reaction enzymatic reaction is first option, that is transient but stable, transient but stable. B option, permanent but unstable, permanent but unstable. C option, transient and unstable, transient, unstable. And D option is, that is permanent and stable, permanent and stable. So it is very important here at the time of enzymatic reaction we have discussed this thing. So this is really transient conditions, transient and really unstable. So the correct answer is transient and unstable. So next question is question number 13. In 13, the essential chemical component, the essential chemical component, essential chemical components of many coenzymes are, many coenzymes are first option, that is proteins. B, that is nucleic acid, that is nucleic acid. And C option is, that is carbohydrates, and D option is vitamins. So essential components of many coenzymes we have discussed in the theory classes of previous videos. So vitamins plays very vital role for, and what niacin we have also discussed, that is, so these vitamins plays vital role and the components of coenzymes. Then question number 14. A phosphoglyceride is always made up of, a phosphoglyceride is always made up of, first option, only a saturated fatty acid, only a saturated fatty acid, only a saturated fatty acid esterified to a glycerol molecule, to which a phosphate group is also attached, a phosphate group is also attached. Then the second option is, that is only an unsaturated fatty acid, only an unsaturated fatty acid, only an unsaturated fatty acid esterified to a glycerol molecule, to which a phosphate group is also attached, a phosphate group is also attached. And the C option is a saturated or unsaturated fatty acids, a saturated or unsaturated fatty acid, unsaturated fatty acid, saturated or unsaturated fatty acid esterified to a glycerol molecule, to a glycerol molecule to which a phosphate phosphate group is also attached, and the D option is a saturated or unsaturated fatty acid, a saturated and, or we can write saturated or unsaturated unsaturated fatty acid unsaturated fatty acid esterified to a phosphate group which is also attached to a glycerol molecule. So the important thing is phosphoglycerides is always made up of, so there should be the presence of either saturated or unsaturated fatty acids esterified to a glycerol molecule, and phosphate group should be attached. So the correct option is a saturated unsaturated fatty acids esterified to a glycerol molecules with a phosphate group is attached. So here we can draw the structure: CH2OH, the trihydroxy alcohol, the glycerol, and the fatty acid should be there, and the phosphoric acid phosphate group should be attached. So this is the correct option. And next, the last question is the question number 15. Macromolecules chitin is first option nitrogen containing polysaccharides, B phosphorus containing polysaccharides, and C option is sulfur containing polysaccharides, and D option is simple polysaccharides. So this thing we have already discussed. This is the macromolecule, large molecule chitin, and this chitin is made of N-acetyl glucosamine, where the amine group or nitrogen containing polysaccharide is the correct answer. N-acetyl glucosamine. I have already discussed the structure also, the amine group and the acetyl group is there. So this is all about the part 3. Then we will discuss in part 4.